Good afternoon to all. I'm R. Madhumita, 3rd BSc Microbiology. Today we are going to learn about the topic pBR322 is under the recombinant DNA technology paper. Now, plasmid. What is a plasmid? Plasmid is an extra chromosomal DNA, small circular and double-stranded DNA molecule that is distinct from a cell's chromosomal DNA. Plasmids are naturally exists in bacterial cells and they also occur in some chromosomes, eukaryotes. pBR322 is an artificial plasmid which is usually regarded as the boon for gene cloning techniques. pBR322 is a plasmid and widely used in E. coli cloning, created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. It was named after Francisco Bolivar Zapata, the postdoctoral researcher, and Raymond L. Rodriguez. Its DNA is derived from the different but naturally occurring plasmids. The DNA sequence of entire pBR322 has been mapped and published. Many other plasmids were artificially constructed to create one that would be ideal for a cloning purpose, but pBR322 was found to be most versatile by many and was therefore one of the most popularly used. A large number of other plasmids based on pBR322 have since been constructed specifically designed for a wide variety of purposes. Examples include the PUC series of plasmids, most expression vectors for extra chromosomal protein expression, and shuttle vectors contains the pBR322 origin of replication, and fragments of pBR322 are very popular in construction of intra-species shuttle or binary vectors for targeted integration and excision of DNA from chromosomes. We go to the structure of the pBR322. Structures combined with the origin, sites and selectable marker. Now we come to the sites. What are the sites are present in the pBR322? The plasmid pBR322 has restriction sites for over 20 restriction endonuclease enzymes, and it contains genes that give resistance against two antibiotics, namely penicillin and tetracycline. Cloning in pBR322 at any of the sites within these two genes will result in insertional inactivation of either of these genes. Around 40 different restriction sites are present on the genome of pBR322. Almost 11 different restriction sites are present in the region of tetracycline resistant region. In the ampicillin resistant region contains 9 restriction sites of different enzymes are present. Some of the known restriction enzyme sites are BAMH1, HIND3, EcoRI, SAL1 or many more. Recognition sites are the genetic sequence where the restriction enzymes cut the DNA segments. Selectable marker sites or two selectable marker sites are antibiotic resistant genes are present of genome of this plasmid ampicillin resistant site. The ampicillin gene codes for beta-lactamase, which can be used for screening microorganisms when a foreign DNA is being inserted in the plasmid. Tetracycline resistant site, this gene degrades the antibiotic tetracycline and can be used for screening microorganisms. These antibiotic resistant genes are useful in screening organisms after cloning. Unique selectable marker sites within the gene. Ampicillin resistance are such as PSI1, PBU1, SCA1. Tetracycline resistance are BAMH1, BSPM1, EcoRV, NHEL, NRULE, etc. Selectable markers are play a supportive role in the differentiation of transformed cells from non-transformed cells. They also aid in the distinguishing between recombinant and non-recombinant cells. Now we see about the advantages of pBR322. The plasmid pBR322 is most commonly used plasmid in gene cloning experiment. It becomes an ideal cloning vector due to the following reasons. This plasmid is much smaller than a natural plasmid, making it more resistant to damage by shearing. Small size of the pBR322 also increases the efficiency of uptake by bacteria during transformation. Since the plasmid of pBR322 is small sized, so foreign DNA up to 6 kb in length can be inserted into this plasmid. Next, the plasmid pBR322 has relaxed origin of replication. Multiplication of this plasmid is not tightly linked to cell division. So, in the presence of this antibiotic chromophenicol, the number of pBR322 may increase to 1000 to 3000. The plasmid pBr322 is used as a raw material for the creation of several other useful plasmids or other cloning vectors. Next, disadvantages. Mainly disadvantages comes below the pBr322 was instability. The plasmid pBr322 is lost in continuous culture in the absence of selective pressure. This may create problems for the large scale fermentation of recombinant bacteria for investigate or commercial purposes. Copy number. The copy number, because of its relaxed mode of replication, the plasmid pBr322 is present in approximately 20 copies per chromosomes. This multi-copy feature can produce a gene dosage level that may be too low or too high for a particular experimental condition. Availability of cloning sites. pBr322 has 21 unique restriction sites, but only 11 sites can be used to insertionally inactivate the antibiotic resistant genes. Another 3 unique sites fall within the origin region and therefore cannot be used for the cloning purposes without impairing plasmid replications. The screening process time consuming and laborious. Now we saw about the construction of pBr322. The origin of the plasmid. What are the origin of the plasmid? The plasmid pBr322 was constructed by using the 3 different naturally occurring plasmids. Thus, the gene for ampicillin resistance was derived from RSF2124. The gene for a tetracycline resistance was taken from PSC101. The origin of replication was obtained from PMB1, which is related to the plasmid col-E. The copy number of this plasmid is 15 to 20. The origin of the plasmid pBr322. The boundaries between the PSC101, PMB1 and RSF2124 derived material. The numbers indicate the positions of the junctions in base pairs from the unique eco-RI site. The molecular origins of the plasmid pBr322 RH768 was isolated in London in 1963 and later renamed R1. First, a variant R1DRD19, which was depressed for mating transfer, was isolated. The ampicillin resistant transposition from this plasmid was transposed onto PMB1 to PMB3. This plasmid was reduced in size by eco-RI rearrangement to form a tiny plasmids. PMB8, which carries only a collagen immunity. Eco-RI fragments from PSC101 were combined with PMB8 open and unique eco-RI site, and the resulting chimeric molecule rearranged by eco-RI activity to generate the PMB9. The separate event, the TN3 of RNDRD19 was reposed by col-EI to form PSF2124. The TN3 element was then transposed to PMB9 to form pBr312. Eco-RI rearrangement of pBr312 lead to the formation of pBr313, from which 2 separate fragments were isolated and ligated together to form the pBr322. During the series of construction, R1 and col-EI served only a carriage for TN3. Now the construction. Although PSC101 and col-EI and RSF2124 can be used to clone DNA, suffer from a number of disadvantages as outlined above. For this reason, considerable effort has been expended on constructing in vitro superior cloning vectors. Undoubtedly, the most versatile and widely used artificial plasmid vector is plasmid pBr322. It contains the ampicillin resistant and tetracycline resistant genes of RSF2124, PSC101 respectively, combined with replication elements of PMB1. Plasmid pBr322 is the most widely used cloning vehicle. In addition, it has been widely used as a model system for the study of prokaryotic transcription and translation, as well as the investigation of the effects of topological changes on DNA conformation. So from this video, you got an idea about the plasmid pBr322 and its constructions. I think this video is very useful to your learning purpose. Thank you.